Hi everyone. Uh, we are just approaching a wonderful topic in the molecular biology, especially for the class 12 of the NCERT textbook. So we are going to just examine what's called DNA packaging. Now, studying this for examination and getting marks is one thing. That is perfectly all right. You have to get marks. But understanding the molecular phenomena that happens in a small cell and inside the nucleus, that too, where the molecules play such a drama that it is impossible for any one of us to understand that completely. Researchers have been trying from 50 to 60 years to understand what is really happening in the nucleus, especially for the DNA molecule. It is very difficult to examine that. The moment you understand that, the moment you feel that it is, you are going to understand that, you will jump with joy, because each day maybe about 30 million cells are produced in the human body and destroyed. You can imagine the amount of mechanism, the amount of activity, the amount of molecular drama, proteins joining, then the enzymes working on that, extra molecules coming into that one, how they get into the cell, how they become adjoining with one another, what even the minor chemical changes, a charge, positive or negative charge, would change the whole drama on the situation. And such a thing happens in what's called DNA packaging. So what we do, we will try to quickly know what do we mean by DNA packaging. So DNA packaging refers to it is an arrangement by the cell in which nearly about 2 meters of the DNA molecule in the resting cell, that is the normal cell, will be there in the nucleus. That's nearly 10 nanometers across diameter. That is the thing. So that means 2 meters. If you expand 2 meters, we'll find out how to find the length. 2 meters of the DNA molecule will be completely packed inside the nucleus, and they will duplicate themselves into form double that, and then they divide into two cells. Qualitatively and quantitatively, the genetic material is passed on. So packaging refers to the packaging is nothing but compacting or condensing or making it smaller and smaller. If you have a very large molecule like that, then it will be compressed into coils and coils like that, and ultimately they will be compacted into chromosomes. That's called the packaging. And why packaging is required? After all, if you ask a question, the cells are not very simple things. There are enzymes which can break the molecules in order to see the genetic material contained in a cell goes to the cells equally and qualitatively. That is, both in quality and quantity, the progeny or the new cells must get from the parent cell, and to ensure that there is a packaging, and that is the importance of packaging. So packaging to ensure genetic material is shared with daughter cells in quality and in quantity. That is very important. You can just remember, if you just get a little injury on your skin, in no time, maybe in about 4 to 5 hours, or in very young children, maybe little longer time in the older people, will be replaced by the skin cells. Skin cells are replaced only by skin cells, not by muscle cells. When the muscle is torn, only muscle cells are made and replaced, not the bone cells. So like that, the cell has a mechanism and intelligence in it to make the things. For that, packaging is a must. And then how do we calculate the now 2 meters? How did we come to this point? It may be now 2.2 meters, they say. How did we come to that kind of a number? If you say, normally what you do is number of base pairs in the DNA molecule. See, people have done a lot of research and they have given us an estimation that the number of base pairs in different organisms. So we will now just take up the mammalian cell. Mammalian cell may have a large number of nucleotides in that one, and they will form into base pairs. And it seems the base pairs in the human cell will be to the order of 6.6 raised to the power of 10 raised to the power of 9. That means that will be about 66 billion cells. I mean base pairs. Base pairs will be there. And distance between the base pair will be, supposing if this is a base pair, you take one base pair, second base pair, distance between these two will be in nanometers. That will be 0.34 nanometers. Nanometers are expressed in the like 10 to the power of minus 9 because they are much. That will be 1 billionth of a meter. I mean, it is 1 billionth of a meter is called the nanometer. So that is the smallest part we can think of. So the length of the DNA molecule is calculated by the number of base pairs in the DNA and the distance between the base pairs. That approximately comes to 6.6 raised to the multiplied by 10 raised to 9 and multiplied by 0.34, that is the distance between the base pairs, multiplied by 10 raised to minus 9. That approximately gives 2.2 meters in length. That is the length of the DNA molecule. Now the question comes, or you must ask a question, how that little cell can do activities to make it smaller and smaller and smaller and smaller. 2 meters will have to be packed in a small nucleus, and that's how it happens. Let us try to know how it happens quickly. Now, since my blackboard is very small, I have to rub it off all the time and then rewrite it. So what I do now will be that I will try to just say that the research made by various scientists using X-ray diffraction, using ultraviolet say magnification of the molecular structure, and then ultrafiltration, electrophoresis, crystallography, and using electron microscope. So all these studies they have done, and ultimately they have come to a conclusion that the DNA molecule, which is a long one, will first get into a kind of a structure like this, like what we call, it's called the string and the bead type of a formation. This would be the first type. The Korenberg, a scientist, suggested in 1976, he suggested there will be a kind of a arrangement like this. Not only one, two more scientists also suggested the same thing, but Korenberg ultimately said the DNA molecule will get itself into a kind of a bead on string structure. So this is the first thing that happens. What is that really happens here is explained further by means using the concept called the nucleosome. So the first organization will be DNA get itself organized into nucleosomes. In the human mammalian cell, it seems about 30 million nucleosomes are possible. Now let us try to say what is this nucleosome, how the nucleosome is made. We will try to see. For the sake of reference, I have made a small presentation here, a diagram which you can see here. All of you can see here. See here, I have taken the, you can see this one. This is the DNA structure. DNA gets into, but the DNA becomes a nucleosome that will contain 8 histones. And these histones are called H2A, H2B, H3, H4. That is, 4 of them will form 8 histones, and then one more will become, they will become the octamer. That means now there will be 8 histones, and these histones will, I will rub it off now so that we will be able to see the things structure properly. So there will be, say for example, 1, 2, 3, 4, 5, 6, 7, 8. So there will be 8 histones. Histone is a protein. Histone is a protein, and this will be having, there are 2 types of say components in that. One is arginine, second is lysine. So these 8 pieces of histones, that will be H2A, one more H2A will be there, H2B, H2A, H2B, like that, H3, and then H4, like that. So the 8 of them, that is, will form what's called the octamer. 8 histones will form the octamer. That is the first thing. They are rich in arginine and lysine, and that gives these histones a slightly positive charge on them. There will be positive charge on them. And now the DNA molecule, the DNA molecule, now for example here, DNA molecule might, that is the DNA molecule, that will come and binds itself on this one. They will bind on that one, and then they will form one more round. That will be like that, the DNA will come out. So the DNA enters here and the DNA comes out, and it makes approximately 1.75 round. This will be around that one. And that will be, that means the histones will be inside, and outside the DNA binds, and histones will be positive in electrically and DNA will be negative electrically. So their binding takes place, and that will be, then in biochemistry you can study more of these things. Here it is not possible to explain that. So they bind like that, and then in order to see this will not go away, there will be one more histone that joins here. I will use a different color so that you will understand. This is another histone called H1. That is one more histone, and this histone binds the DNA molecule that comes inside and that goes inside. And the scientists have suggested that this two circles that goes on on the DNA when the binding takes place, that will contain 200 base pairs totally. This is one nucleosome. Now this structure with 8 core, they are called the this is the octamer, and the DNA surrounds that. That DNA is called the core DNA, and then the outside, the DNA that comes out, this joins with another nucleosome, that's called the linker DNA. So now scientists have said that this total structure will contain 200 base pairs, out of which there will be 73 base pairs in one circle and another 73 base pairs in the second circle. That makes 146 base pairs. The rest of the base pairs, that will be nearly 4. Nearly 34 base pairs will be in the linker DNA. So this is structure, and that structure is called the nucleosome. So such a DNA and histone complex is called the nucleosome, and such nucleosomes are in plenty. Now this figure again I will show you. This is the nucleosome forum, this is the nucleosome, and such nucleosome get connected by means of this linker DNA, and again they will form, linker DNAs will form, and nearly 8 such nucleosomes will form a core, and that core will be called by name solenoid. It becomes solenoid, core-like structure, and that core further coils and further bending takes place, and ultimately they will come to chromatids. So the DNA packaging starts from the interface and goes on the whole drama in forming the nucleosome and other things, and ultimately end up in the chromosome. That is the DNA packaging that takes place. Now there are a number of things that one could learn from these things if you know exactly how the DNA packaging goes on. I think I will stop at this part of the time, and I hope you understood that here. The most important thing you must know is that there will be the DNA will form the nucleosome. Nucleosome is a complex made out of histones, 8 histones in the center, and they will come together, and then the DNA coils around that to 1.75 coil, and each coil will contain 73 base pairs, and then the small one will be linker DNA that will be connected to other thing. So this is about the DNA molecule, how the compacting, how the packaging takes place. There are so many things to learn on this. I think if you just understand 2 things, one, the meaning of the term nucleosome, you must be able to explain that, and then if you are asked to find out how do you find out the length, then you must use the formula. That formula will be length of the DNA is equal to number of base pairs multiplied by length of base pair, that is between the base pairs, length between the base pairs. So if you multiply these two, you get the length. So that is the one you must remember. So in the examination they may give you, now the number of base pairs in the human mammalian cell will be 6.6 multiplied by 10 to the power of 9 is the order. You may get about 66 billion cells. That will be multiplied 66 billion base pairs. That will be multiplied by a distance 3.4 nanometers. No, sorry, that will be one full thing. That will be 0.34 nanometers multiplied by 10 to the power of minus 9. That is the smallest one. So if you multiply these two, you may get 2.2 meters approximately. So this one maybe you may have to remember for the examination, this formula. So if you remember that, you will be answering a question on the nucleosome. Thank you.